So Tom, you've been here for a while, and I'm sure that many have come calling to try to snag you to other places. What's kept you at Stanford, and what makes you excited about being at Stanford?
I think there are a lot of great positives about Stanford. When I left Vanderbilt a couple of decades ago, my chair of medicine at the time said, "Why would you leave Vanderbilt? Why would you go to Stanford?" And I said, "I'm going to Stanford to do human genetics," and that's basically what I've done. And so I've collaborated with investigators in the Department of Genetics, bioinformatics, biostatistics, and it's been fabulous to be able to work with some great investigators in these other departments and push human genetics at Stanford. We contributed, I think, a great deal to the genome-wide association studies investigating genetics of metabolic and coronary artery disease traits, and I think the community at Stanford is really fantastic, and so that's really been the thing that has kept me here more than anything else. Yeah.
There's something else that I've admired about you is just taking advantage of the entrepreneurial spirit at Stanford. So you've helped form a few companies, and that seems like a pretty unique aspect. Tell me how you've taken sort of advantage of those opportunities.
So when I came to Stanford, I sort of looked at that entrepreneurial environment and I said, well, that's great, but it's not going to involve me. I'm never going to be part of that. I'm just going to stay in my lab and, be chief and do my research and keep my NIH funding solid, and I'm not going to be distracted. And then one day, one of my postdocs came and sat on my couch and said, we've been trying to diagnose things, by looking through a microscope and staining tissues with plant dyes, and now we have this capability to do genomics and genetics, and we ought to really be able to do a much better job. And I said, okay. And so we started a small company in a warehouse in South San Francisco to use genomics to better diagnose transplant rejection, focusing on heart transplantation, rejection of transplanted hearts. And so that went on to become, the genesis for the LMAP test, which is commonly used today for judging rejection for patients. And so that's been fantastic. And it wasn't a distraction. It was something that brought me great joy and translated the work that we were doing, looking at gene expression, translated that into the clinic. So for me, that was fantastic. And so I think it's been a great success, and just to see that translation aspect of the work has been really very rewarding. So
what is your leadership or mentorship style? How have you spawned these folks coming here and then blossoming and moving other places?
Well, I've been very fortunate to have some very talented fellows come and work in my lab, and so that's been first and foremost, I think, and it's been easy to help very smart young folks who are very focused on doing academics and research, to help them make the transition into a faculty position. I think the important thing for me is to value their time and to take their participation in the lab work to a level where I feel like I'm very much I feel like their career is in my hands, and so it's my job really to help them learn the things they need to learn and to make the correct choices and avoid the bad choices and to develop a plan for themselves that will allow them to conduct good research, to learn the basic instrumental techniques and things that they need, and then just how to think about science. So I think that's really been very successful for me. And I have a family now. I have my own family at home, but I also have a family of trainees that have worked with me around the United States and indeed around the world. And so I see that. And so I continue to be a mentor,
Yeah.
Even after the fellows go on to their independent career. And so I see that as one of my major roles. And derive great satisfaction from that.
I know that two of our outstanding fellows, one of whom is graduating, Rob Worka, he's going off to a faculty job elsewhere, and also Paul Chang, one of our current um, fellows, soon to be faculty members, part of the translational investigator program. Tell us a little bit about the research they've done with you and what you're doing in your lab now, and where you hope to be going in the next few years.
I think Rob is a great example of what a fellow can achieve, I think. Rob was really pretty uncertain about what his career path was going to be, and if he was going to do research, exactly what type of research he was going to do. And so he and I began to have a series of meetings every month for a few months, and I sort of outlined for him the basic directions he could go. And then I told him to go sit on the beach for a couple of days. And indeed, he took his wife and his kids and went to the beach and thought about it. And I said, you know, you really have to decide what's important for you and what is exciting for you. And so he came back after all that. And he said, well, I've decided, I know what I want to do. I know exactly what I want to do now. And I said, okay, tell me. He said, I want to come and work for you, and I want to understand the genetic basis of coronary artery disease, and I want to really, you know, use cutting edge tools to move the field forward. And so he knew virtually nothing about bioinformatics infrastructure, about computing, and he's gone on to learn how to incorporate the latest tools and bioinformatic approaches into his research, and has developed in my lab the single cell RNA sequencing and single cell attack sequencing protocols that really allowed the lab, I think, to move to the forefront now and to be able to characterize the genes and the mechanisms and epigenetic mechanisms that regulate gene expression. So I think he has catapulted the lab really to the forefront of the research that needs to be done now to follow up on the human genetics studies. So he's a great example, I think, of what a fellow can achieve in 3 years. It's been remarkable.
Maybe talk a little bit about, you know, the importance of Stanford in the global climate of human genetic research and cardiovascular disease, and some of your, you know, prouder achievements in that era, and where you think we need to be going in the next 10 years.
Well, I think I think that every decade or so there is a fundamental breakthrough that allows progress to be made in one direction or another, and cloning of genes, for instance, was a fantastic breakthrough that allowed the characterization of genes that regulate fundamental physiology and pathophysiology. Without a doubt, the past decade has been an unprecedented time for the mapping of human genes, and we're ready now to have the human uh, be a model system for us to study human disease, and it's been just a fantastic opportunity. Uh, it's important now that we follow up the human genetic findings and we try and understand what are the mechanisms of these associations. What's the causal variation? What's the causal gene? What does that gene do under normal circumstances? What does it do under a pathophysiological circumstance or condition? And um, and then without question, I think that type of research will lead us to understanding pathways which are fundamentally involved in the disease process, and then we can develop targeted therapies to the blood vessel wall. We have no drugs which treat atherosclerosis currently. We have drugs which treat risk factors. But very soon, I think in the next few years, we will understand what are the targets that we can go after, that we can develop drugs that will actually target the disease process. So I'm very much looking forward to that. I think there's uh, a decade, 2 decades, or 3 decades in front of us now where we will try and apply this information looking at risk assessment and trying to integrate the genetic risk assessment into the practice of medicine. But at the same time, you know, as I was saying, we'll be able to use that information, and probably it's going to be more important what we can do with that information to develop new therapies. So I think it's an incredible time to be involved in this field. I'm so happy that this has happened at this point in my career, and I've been able to take advantage of the new findings to really better understand and to focus my research on these critical pathways. because before we knew there were a lot of changes in the blood vessel wall in terms of the number of cells that were there, the types of cells, genes that were being expressed there, but we didn't know what was causal. And so now we use allelic variation in the genome, which is Mother Nature's way of perturbing the system, so that, and we can now characterize those perturbations, and we can understand and we can assess causality, which we could never do before. So now we know the pathway, we know the genes, we're learning the pathways, we know that the things we're studying are causal. That has changed everything.
When you came here, there was 25 faculty members, and now there's close to 80. The research mission and the clinical mission has also grown. Tell us a little bit about your proudest achievements as a division chief.
It's all about the faculty, I think, and bringing in, really exceptional faculty in not only the basic science and genetics, although we've grown tremendously in that area, and I think we'll probably have the strongest program in the country now, but in electrophysiology, in imaging, and so lots of areas we created some growth that I think allowed us to build fundamental programs and vertically integrated structures where we would have great basic science, great translational science, and great clinical care being delivered. The Amyloid Center, for being one great example, I think, where we've got the best basic science, the best translational research, and fantastic clinical care. And so I think that's been really, it's all about the faculty and building programs. I think that has been the most rewarding thing that Alan and I have done while we were chiefs at Stanford. And it's been a pleasure to hand that off to Eldrin and see him, begin to take the reins and push the division forward. So I'm really happy to have done the things I've done, and I'm perhaps more happy now to have done it and moved back to the lab where I'm going to push the science, with and now I can put 100% of my effort into that, and so I'm very excited about that.
I've asked the questions that I wanted to ask. Is there anything else that you wanted to say?
I think the division has grown. It is more complex than it was when I came here. I've accomplished a lot of the things I wanted to do as division chief. We have the largest and probably the best inherited cardiovascular disease center. We have thousands of terabytes of data, on various genetic studies, human genetic studies, and we have a lot of capability now. And so I think it's time my role is clear in the division now. I want to train more fellows. I want to do more research. And it's great to just be here. And as you say, I think it's a great environment. I think people work very well together. It's the most collaborative place I've ever seen. And so I think that's one of the great strengths. And I'm just happy that things have gone well, and I'm happy to still be here, and I'm happy to be doing my research.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.